Welcome back, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to resume the chapter 18, that is body fluids and circulation. In the previous lecture we have already covered the components of blood. We have seen introductory part of this chapter. Today we will see about the blood groups. Okay. So, blood of human beings differ in certain aspects, although it appears same in all individuals, right? So, when we are talking about the blood groups, right, the blood of human beings it differ in certain aspects. So, even though it is the contents of the blood is same, that is the cell as well as the plasma, right? But still, there are some aspects, right? And due to that, the individual's blood is different, right? So in the blood grouping system, right, we are going to see two main types of grouping. First is ABO grouping system or blood group system, and second is RH grouping system. So first we will see about ABO grouping system. So ABO grouping is based on the presence or absence of two surface antigens, RBC, that is antigen A and antigen B. So the ABO grouping system, this is based on the presence or absence of the two surface antigens which is present on the RBC. Okay, and those two surface antigens are antigen A and antigen B. So you need to keep in mind that on your RBC there is presence or absence of antigens, right? So and those two antigens, either maybe there can be presence of antigen A or antigen B, or there can be absence of none of this, right? So neither antigen A is present nor antigen B. So the plasma of an individual contains two antibodies produced on in response of antigens. So see, the blood is composed of two things. First is the blood cells and second is the blood plasma. So your blood plasma contains two antibodies, right? Which is in response with the two antigens which are present on the RBC, right? So the components of blood, that is blood plasma and blood cells. The blood cells contain the two antigens, that is antigen A and antigen B. And your blood plasma, it contains two antibodies, that is the that will be in response with the antigen, right? So now we will see that what exactly how the different types of blood group are formed. So as you can see in this table, there here it is written four different types of blood group where I'm showing the cursor, right? So A, B, AB and O, right? So we will see that how the blood group of an individual is being selected. So let's talk about the blood group A. So the person who has the blood group A have the antigen A on its RBC, and there will be presence of antibody B in the plasma. Now see, if antibody A is present in the plasma when the antigen A is there, then they cannot bind with each other, right? So, sorry, they can bind with each other, right? So for this reason, the RBCs can die because the antigen is present on the RBC only, right? So the killing should not take place of your RBC, and for this reason, when the antigen A is present in that person, the antibodies will be anti-B. So the donors group, that is who can donate the blood to this type of individual who is going to possess the A blood group, right? So that is again the donor group will be A and O, right? So this two can donate the blood to the person who is having the blood group A, right? Second is B blood group. So in B blood group, the RBC is going to contain the antigen B, right? And here also there will be opposite antibody which will be present. So that means the anti-A antibody will be present in the plasma. And here again the donors will be B group and the O group, right? So these two are going to do, these two can donate the blood to the person who is having the blood group B. Third is AB blood group. That means here both the antigens are present. That is, even A antigen is there as well as B antigen is also there. But there will be no presence of any of the antibodies in the plasma, right? Because if there will be presence of antibodies, then it can bind and it will kill your RBC, right? So for this reason, when both the antigens are present, none of the antibody is present. And who can donate the blood? So that is the person who is having AB blood group, A, B and O. So all can donate the blood to AB blood group individual, right? And last is O blood group, right? So that means there is presence of no antigen on the RBC. So the person who is having O blood group, this person is not having any of the antigen on the RBC, but they possess both the antibodies in the plasma, right? So there will be presence of antibodies, but not antigens, right? And who can donate the blood? Only the O group can donate the blood to the O blood group, right? So this way, the based upon the antigens on the RBC and antibodies in the plasma, the blood group of an individual is decided. Now, during the blood transfusion, blood of donor has to be matched with the blood of recipients to avoid clumping of RBCs, right? That means when the particular blood transfusion is taking place at that time, initially the donor's blood as well as the recipient's blood it should be matched, right? And if the matching is taking place, then and then only the transfusion should take place, or else the clumping of RBCs, that is, they form the clumps, big clumps, and this way the person may also die, right? The group O blood can be donated to any individual with any blood group, so it is called universal donor. As you know that here you can see the donors group. In donors group, every column, right, here it is O group. That means the person or the individual who is having O blood group, it can donate to any individual with any blood group. For this reason, as that person can donate its blood to any of the individual, for this reason that blood group O is known as universal donor. And the person with AB, here you can see that this AB, the donors group, it contains all the group, right? For this reason, as it can receive the blood from all the blood group, for this reason, this particular blood group is known as universal recipient. So this is all about the ABO grouping system. Now we are going to see the RH grouping system, and this is based upon the RH antigen, which is similar to rhesus monkey. This was first observed in the rhesus monkey, and this RH antigen is been observed on the surface of RBCs of majority of individuals. That means most of the people is having RH antigen on their RBC. So around 80% of the people is having RH antigen on their RBC, right? And those people who are having the RH antigen on their RBC, those people are called RH positive. And the ones who does not have this antigen on their RBC, those are known as RH negative, right? So the people with RH antigen, they are said to be RH positive, and the people who does not have RH antigen on their RBC, those are known as RH negative, right? Now there is a erythroblastosis fortilis, right? So this is a state, or this is a case in which the father's blood is RH positive, right? So if father's blood is RH positive and mother's blood is RH negative, then the fetus blood is going to be RH positive, right? So as you can see over here, this is a condition which is observed, in which when the father is having RH positive and the mother is RH negative, then the fetus blood is going to be RH positive, because the father's blood is passed to the mother's body. So sometimes what happened, for the first pregnancy it will be normal, But during the delivery of first child, there is possibility of exposure of mother blood with fetus blood to develop antibodies in mother blood. So here what happens, as the Rh positive for mother is going to be foreign thing, right? That is, it is not mother's own body, right? For this reason, the father's blood is when passed to the mother's blood. So during the first delivery, what happens, the baby's blood, right? So the baby's blood is exposed to the mother's blood, and for this reason, what happens, the mother's blood is going to develop the antibodies against the Rh positive antigen, right? Because the father's blood was having Rh positive and the mother's blood was containing Rh negative. So the mother's blood is only knowing about the Rh negative one, right? So when the child's blood is exposed to the mother's blood, at that time the mother's blood is going to take up the Rh positive as a foreign particle or a foreign antigen, and for this reason, the mother's body is going to develop the antibodies against the Rh positive antigen. So when the in the subsequent pregnancy, that is when again that lady or again the mother is going for the pregnancy, right? So there can be chance that the mother's blood can leak into fetus blood and destroy the fetus RBC. So here what happens, in the second pregnancy, after the first pregnancy, if the injection related to the antibody development is not done, at that time what happens, there are chances that in the second pregnancy the mother's blood is already having the antibodies of the Rh positive antigen. So from the mother's blood, those antibodies are going to pass to the baby's body, and the fetus RBC is going to be destroyed. And this can lead to a case which is known as erythroblastosis potilis, right? So that is erythro, that is RBC, blastosis, that is the blast is taking place, that is rupturing or destroying takes place of the fetus, and for this reason this case is known as erythroblastosis potilis, right? Now we are going to see about the next topic, and that is coagulation of blood, which is simply known as blood clotting, right? So when an injury is caused to a blood vessel, bleeding starts, which is stopped by a process called blood clotting. So if you are having any cut, right, so when an injury is being caused, at that time your blood vessel is cut, and for this reason the bleeding starts, that is the blood flows from inside to outside, right? So the process in which the blood is stopped and not being flowed out, at that process is known as blood clotting. So an injury or trauma stimulates the platelets in the blood to release certain factors that activate the mechanism of coagulation. So as I said, that thrombocytes, that those are platelets, they are responsible for stimulating, that is for activating the process or the mechanism of blood clotting, right? That how they are doing this, let's see. So calcium play a very important role in blood clotting, right? So let's see how calcium is very much important. So here this flowchart explains that when an injury or removal of blood vessels from removal of blood from vessels takes place, so at that time several steps are being taking place in order to clot the blood, right? So when an injury is being occurred, so at that time what happens, there is presence of prothrombin, right? So in the presence of calcium ions, right, with the help of the prothrombinase, right, this prothrombin gets converted to thrombin. So the first preliminary step is the conversion of prothrombin to thrombin with the help of the calcium ions in the presence of the prothrombinase enzyme, right? Now this prothrombin is getting converted to thrombin. After that, the second step is the conversion of the fibrinogen to the fibrin threads plus the blood cells and plasma. So this thrombin is responsible for activating the fibrinogen, and this fibrinogen upon activation gets converted to fibrin threads. So it is going to form the thin fibrin threads, and along with those fibrin threads, the blood cells and plasma are also formed. So here first step says that the prothrombin gets converted to thrombin within the presence of the prothrombinase and the calcium ions. Now this thrombin is responsible for activating the fibrinogen to fibrin threads as well as the blood cells and plasma. So when the fibrin threads are formed, and they form the network-like structures which does not allow the inner blood to flow outside the body. And this forms a clot. So this way the blood clotting mechanism takes place, right? So now we are going to see about the lymph, right? So during flow of blood through capillaries, some water soluble substances move out in the space between the cells of tissue. So whatever, the lymph is also known as tissue fluid, right? That is, whatever intercellular spaces are present in between the tissues, that space is going to contain a fluid which is known as the lymph, right? And they flow during the flow of the blood through the capillaries. What happens, some of the water soluble substances, they move out of the space and they enter into the tissue fluid which is known as lymph. So this fluid released out is called interstitial fluid or the tissue fluid. So whatever fluid is released out in between the cells of the tissues, that is known as interstitial fluid, or we can simply say as the tissue fluid. Now this is similar to the blood but has fewer blood proteins, less calcium and phosphorus and high glucose concentration, right? This lymph is almost same with the blood, but it does not have the same proportion of blood cells, right? Or the blood proteins. They are having less calcium than phosphorus, and they do possess higher glucose concentration, right? That means the glucose concentration in the lymph is also more, right? This lymph is colorless fluid and it is going to contain specialized lymphocytes that provide immune response to the body. So this lymph is also one of the important body fluid because it is colorless in nature and it is going to possess specialized lymphocytes. As you know, the T lymphocytes and the B lymphocytes, and these two of them, they are going to provide you the immunity. And this way the immune response to your body is generated. And the main function of lymph is to provide immunity, carry proteins and fat molecules, and transport oxygen, food materials, hormones, etc. So just like the blood, the lymph is also responsible for carrying the protein and fat molecules. Also, it is going to transport the oxygen, then food materials, as well as the hormones, right? And a part of this, the main function of lymph is to provide immunity. How it is going to provide immunity? Because there is presence of specialized lymphocytes in it, which is going to provide you the immune response, right? So this is all about the lymph, which is known as tissue fluid. I hope we are very clear with today's topic and I hope to see you again. Thank you.